Hi friends and dear students, this is Kanita serving as Head and Assistant Professor in the Department of Biochemistry, JJ College of Arts and Science, Autonomous, Pudukkottai, Tamil Nadu, India. In this video, I am going to brief you about the various models for the DNA replication. Before getting into the exact topic, let me explain you about the cell cycle. The cell cycle is a sequence of events that occur in a cell during cell division. It results in the formation of two identical daughter cells, and the duration of the cell cycle varies from cell to cell. The cell cycle comprises of four phases. The first one is the G1 phase, that is the gap 1 phase. Second one is the S phase, that is the synthetic phase. Third one is the G2 phase, the gap 2 phase, and the last one is the M phase, the mitotic phase. The G1 phase, in this phase, the preparative phase for DNA synthesis. All cellular components replicate except DNA. Cell size gets increased, and if any damage is present in the DNA, it is being detected during this particular phase. Following G1 phase, we have S phase, that is the synthetic phase. This is the phase in which the DNA undergoes replication. After the S phase, we have G2 phase, that is the gap 2 phase. This is a phase where the cell prepares itself to undergo cell division and forms the spindle fibers. Any damage to DNA is being detected in this phase also. Last one is the M phase, that is the mitotic phase. This is a phase where the cell undergoes cell division. This phase includes prophase, metaphase, anaphase, and telophase. After mitosis, cell may continue cycle by either re-entering into the G1 phase, or it can enter G0 and remain dormant, or it may lead to the cell death. Besides maintaining the integrity of DNA sequences by DNA repair, all organisms must duplicate their DNA accurately before every cell division. DNA replication occurs at polymerization rates of about 500 nucleotides per second in case of bacteria and about 50 nucleotides per second in case of mammals. Proteins catalyze this particular process. Speed and accuracy are achieved by means of a multi-enzyme complex that guides the process and constitutes an elaborate replication machine. Replication usually occurs in a particular direction only, that is from the 5' end towards the 3' end. And the replication takes place simultaneously on both the strands of the DNA. Replication is hence bidirectional. Replication obeys the base pair rule, that is, adenine always base pairs with thymine and cytosine base pairs with guanine. So on the template strand, if we have adenine, in the newly synthesized strand, thymine gets added up. Similarly, if in the template strand we have cytosine, the guanine gets added up in the newly synthesized strand. Replication results in two daughter DNA strands. Each daughter DNA strand has one parent strand and one complementary strand synthesized newly. Hence, this replication is semi-conservative. Three hypotheses had been previously proposed for the method of replication of DNA. In the semi-conservative hypothesis proposed by Watson-Crick, the two strands of a DNA molecule separate during replication. Each strand then acts as a template for synthesis of a new strand. The conservative hypothesis proposed that the entire DNA molecule acted as a template for the synthesis of an entirely new one. According to this model, histone proteins bind to the DNA, revolving the strand and exposing the nucleotide bases for hydrogen bonding. The third one, the dispersive hypothesis, is exemplified by a model proposed by Max Delbruck, which attempts to solve the problem of unwinding the two strands of the double helix by a mechanism that breaks the DNA backbone every 10 nucleotides or so, untwists the molecule, and attaches the old strand to the end of the newly synthesized one. This would synthesize the DNA in short pieces alternating from one strand to the other. Each of these three models make a different prediction about the distribution of the old DNA in molecules formed after replication. In the conservative hypothesis, after replication, one molecule is an entirely conserved old molecule and the other is all newly synthesized DNA. The semi-conservative hypothesis predicts that each molecule after replication will contain one old and one new strand. The dispersive model predicts that each strand of each new molecule will contain a mixture of old and new DNA. So in order to finalize in order to find out which exact model the DNA replication follows, an experiment was carried out, namely Michelson-Stahl experiment by Matthew Michelson and Franklin Stahl in the year 1958, which supported Watson-Crick's hypothesis that DNA replication was semi-conservative. Nitrogen is a major constituent of DNA. N14 is by far the most abundant isotope of nitrogen, but DNA with a heavier N15 isotope is also functional. So, E. coli cells were grown for several generations in a medium containing ammonium chloride with N15. When DNA was extracted from these cells and centrifuged on a density gradient solution with cesium chloride, the DNA separated out at the point at which its density equals that of the salt solution. The DNA of the cells grown in N15 medium had a higher density than cells grown in normal N14 medium. After that, E. coli cells with only N15 in their DNA were transferred to a N14 medium and were allowed to divide. The progress of the cell division was monitored by microscopic cell counts and by colony assay. DNA was extracted periodically and was compared to pure N14 DNA and N15 DNA. After one replication, the DNA was found to have intermediate density. Since conservative replication would result in equal amounts of DNA of the higher and lower densities, conservative replication was completely excluded. However, this result was consistent with both semi-conservative and dispersive replication. Semi-conservative replication would result in double-stranded DNA with one strand of N15 DNA and one of N14 DNA, while dispersive replication would result in double-stranded DNA with both strands having mixtures of N15 and N14 DNA, either of which would have appeared as DNA of an intermediate density. The scientists continued to sample cells as replication continued. DNA from cells after 2 replications had been completed found consist of equal amounts of DNA with 2 different densities, one corresponding to the intermediate density of DNA of cells grown for only one division in N14 medium, the other corresponding to DNA from cells grown exclusively in N14 medium. This was consistent with dispersive replication, which would have resulted in a density lower than the intermediate density of the one generation cells, but still higher than cells grown only in N14 DNA medium, as the original N15 DNA would have been split evenly among all DNA strands. The result was consistent with the semi-conservative replication hypothesis. I hope you might have enjoyed this video. In the forthcoming videos, we will be seeing regarding the rolling circle model of DNA replication. Thank you.